You'll be surprised to know how much information most animals can pick up using just their olfactory system about sex, reproductive state, health that can directly influence their social behavior.
All that just by sniffing.
Yes. We looked at the rodent vomeronasal organ called VNO, which essentially helps the animal detect chemical signals called pheromones in the environment. The organ has a neural pathway that carries these signals directly to hypothalamus or amygdala in the brain, triggering behaviors like fear, aggression, or attraction. We are interested in these neurons that actually sense these pheromones. Originating from a common stem cell population in the organ, there are two different neuronal subtypes here, and I wanted to understand how these neurons differ from each other at a molecular level. So I tried to profile the differences in how they express certain genes. Most of the differences we found are in the genes related to endoplasmic reticulum. This was surprising because in general, ER has no direct role in sensing pheromone. The ER is actually like an assembly line for production and proper folding of proteins. We discovered that this assembly line called ER in these neurons is actually very differently arranged, and it depends on the type of neuron you're looking at. One group of neuron has very high level of membrane packing. It appears like it is a ER stress-like state, which is generally seen in a pathological conditions. So this is very rare in a neuronal system to see. So we asked the question how this specialized ER is achieved in this group of neurons. So we looked at the genes these two group of neurons express, and we found that a protein called canopy-1 is only expressed in one group of neurons which has specialized ER. And when we removed that gene, we found that the animal's behavior changes and the neurons also start to die during their development. We started by asking how these two neuronal subtypes differ, but it turns out one of them evolved to have a specialized ER doing something quite different, suggesting that what looks like cellular stress might actually be part of how these neurons normally function. Yeah.